This is the cell meiosis video starring Laura Solvrak, Mackenzie Patrick, Jana Linval, and Taylor Weeks. Meiosis is defined as a process in which a single diploid cell divides to produce haploid reproductive cells, which are the egg and sperm cells. We will be showing how spermatogenesis in meiosis is performed. The cell outline seen here is the cell membrane of an animal cell. Here are 6 chromosomes, or what will be 6 chromosomes. The cell begins with interphase. This cell is at the end of interphase, so the DNA has already duplicated but has not begun to condense yet. We then enter meiosis 1. Prophase 1 is the first step. In this stage, the nuclear membrane breaks down and the chromosomes condense, connected by centromeres. Homologous chromosomes link as they condense, forming tetrads. This is where crossing over occurs. When the homologous chromosomes pair up, they exchange parts of non-sister chromatids. Crossing over is an important source of genetic variation. The centrioles also migrate to opposite sides of the cell. In metaphase 1, the nuclear membrane is completely dissolved. Spindle fibers line up the homologous chromosomes along the metaphase plate. The spindle fibers further attach themselves to the centromere of each chromosome. Metaphase 1 is also where independent assortment occurs. That is the random alignment of maternal and paternal chromosomes along the metaphase plate, and is also the second important source of genetic variation. Within anaphase 1, the spindle fibers separate the homologous chromosomes. The sister chromatids remain together. Telophase 1 is where the nuclear membrane comes back. During the cytokinesis portion, which closely follows telophase, the cell splits along the cleavage furrow, resulting in 2 haploid daughter cells. Entering the second stage of meiosis, named meiosis 2, the cell begins in what is called prophase 2. Prophase 2 is very brief. The centrioles once again position themselves on either side of each daughter cell. In each of the 2 daughter cells, there are 3 chromosomes instead of the original 6. In metaphase 2, the sister chromatids, tied together by centromeres, line up at the new metaphase plate within their cell. The spindle fibers attach to the centromeres. In anaphase 2, the spindle fibers separate the sister chromatids. In telophase and cytokinesis 2, 2 processes which are so close to one another that they are simply covered both at once, each of the daughter cells forms a nuclear membrane. They each divide along the cleavage furrow again, resulting in 4 evenly sized haploid sperms. And there is cell meiosis.